Hello, my name is Russell Carter, and today we're going to talk about bioreactors. There are different types of bioreactors. There's the industrial scale bioreactors, which are huge, sometimes up to 100,000 liters. And then there's the smaller scale laboratory bioreactors, which is the one we're talking about today. Let me show you some of the parts. These are the detectors, which is pH detector and oxygen or nitrogen detectors. Then there's the motor, this thing here, which operates the stirrer. The purpose of the stirrer is to bring the E. coli all throughout the media so that they can get food and they can grow. And the reason we need to grow E. coli is so that they can express a protein or a gene that we're interested in. That's the whole purpose of the bioreactor, is to grow these E. coli and grow them so they can express our protein, express our genes.
So in order to use the bioreactor, we need cells to grow in the bioreactor. So in our case, we're going to prepare E. coli cells that are stored at -80. So we're going to thaw those cells, and then we're going to add our plasma to them, which is going to then lead to our protein expression. And that, in this case, we are adding polymerase beta to our E. coli. We are then going to heat shock them. After they're heat shocked, cool them, and then we'll be plating them on the LB agar containing ampicillin. This is because in our polymerase beta here, there is ampicillin resistance. This causes only the E. coli that has picked up this polymerase beta to grow, because that is all we want growing in our bioreactor. We are going to inoculate them overnight, which will cause them to grow. And colonies will form from the single cell that took up our polymerase beta on that ampicillin-resistant plate. After that, we will then take colonies from our plates, put them into 200 ml shaker flask, which contains some LB broth, so they can grow in that broth, so we can prepare them to put into the bioreactor. And inside the bioreactor, there'll be another 800 mLs of LB broth, and that is going to be prepared by.
So while our bacteria are in the shaker flask doing their thing, we can come over here and prepare the bioreactor. Now in the bioreactor, we're gonna add 800 mLs of LB broth in preparation for autoclave. Now we're gonna autoclave on the wet cycle, so we have to put these aluminum foils right here to prevent our broth from becoming diluted in the wet cycle. But at the same time, we don't want pressure buildup inside the bioreactor, so we need to put them on fairly loosely. Snug, but loosely, so there's no buildup of pressure to cause an explosion.
We have our bioreactor and our 800 mLs of LB broth ready to go and autoclaved. We're going to take our 200 ml shaker flask that has our E. coli in it, and we're going to add it into here, getting our E. coli to grow in our bioreactor. To do that, we add in our 200 ml shaker flask into here, and then we set everything up so the bioreactor can run. We'll add the motor, making sure to get it tight on there. Make sure all the ports are plugged in so you can get proper readings on what is inside there. Make sure everything is spun down tight and locked up. And then we'll go over to this program over here that's set up to run the bioreactor. We're going to set it to stir and make sure everything else is going to read how we want it to for this, and set everything else up how it is to grow the bacteria that's in there.
Right now, our bacteria over there, they're in the lag phase. This is the growth curve for the bacteria. Right now, they're in the lag phase, and we're waiting for them to reach the log phase. We don't want them to be in the stationary phase, because at that point, there's equal growth and death. And we definitely don't want them in the death phase, because then they're all dying. So, the way we know that they're in the log phase, we gotta test the optical density at 600 nanometers, at a wavelength of 600 nanometers. When the OD 600 is at 0.6, we know that they're in the log phase for this particular type of bacteria. So we keep testing until we see that. Once we see that, once they're at OD 0.6, we add IPTG. Now over here, IPTG is a molecule that removes an inhibitor in this operon. So the inhibitor prevents the operon from producing polymerase beta. So IPTG removes the inhibitor, polymerase beta is expressed. And that's why we need to add IPTG in the log phase so we get maximum expression of polymerase beta.
And 2 to 24 hours is really the optimal growth time for the E. coli to undergo in the bioreactor. So after that time, we're going to take it out and we're going to collect them. To do that, we're going to take 10 ml aliquots, put them into falcon tubes, and centrifuge them down and collect the pellets to be stored at -80. Those pellets can be used in further experiments to get the protein that we have expressed out of those